So we've got our membranes in place. We've got our biological molecules. Now what we're going to begin to see in chapters 13 and 14 are how do we get information from the environment into the cell and vice versa, which is called signal transduction. Most of the time, your messages are not just going to pass from outside to in, and once they get in, go rect and do something directly. But it's going to be an exchange and a handing off like in a relay race. One molecule is going to bind to the next molecule, that molecule. So it's sending the signal via a whole circuit and cycle of molecules. And as we start looking at this exchange of information, we're first going to look at neurons and how neurons are affected by their internal and external environment, and how changing that environment can lead to this neuron doing some very dynamic things. And we're glad that they do, or we simply wouldn't be here. So when we first look at our neuron or any cell, we're going to see that our plasma membrane that is selectively permeable. Now, are we good with what selectively permeable means? Permeable means diffuse materials can pass. Selective means not everything will. So it's because of this great selectively permeable membrane that we're going to have different molecules on the outside versus the inside, or we can have molecules that are at different concentrations on the outside versus the inside. And for our axon, ions are going to be the most important for what we're considering here with this electrical charge that we're going to generate on the surface of our neuron. And so when we look at a neuron that's doing nothing at rest, we're going to see that that neuron plasma membrane is situated such that we have vastly more sodium ions on the outside of the cell, vastly more potassium ions on the inside of the cell. Now, this is a concept that if you get this down, it's going to help you throughout whatever you're doing with biochemistry and cell biology. Sodium at high concentration on the outside. Potassium is almost always doing the opposite thing of sodium. Whether we're talking about a neuron, whether we're talking about kidney. So if you remember sodium is high concentration outside, you automatically know where potassium is a high concentration. And where would that be? The opposite of sodium, so inside. Now, inside the cell, we also have big molecules. Molecules that are anchored in place, such as your nucleic acids, your cytoskeleton. Many of these are negatively charged. So it's because of many of those negative charges inside, the cytoplasm is usually going to be more negative. And outside the cell where the sodium lives is going to be typically more positive. Bless you. Sorry? Sodium high outside. And where's potassium highest? The opposite of sodium, so inside. So at rest, the cell is typically going to be more negative on the inside because of all these negative ions that are trapped in the cell. Sodium and potassium are both positive charge ions.
Yeah, that's right.
That's correct. But nucleic acids, phosphate groups on the nucleic acids, different charged proteins in the cytoskeleton, they're negatively charged, and there's more of them, and they're stuck inside. So that's the principal reason is we look at a cell at rest. Yeah, a cell at rest is going to be more negative on the inside. And since you don't have a lot of those negatives that get out, and all the sodium is out there, it's going to be more positive on the outside of the cell. And when you look at that charge separation, positive outside, negative inside, what does that sound a little bit like? You ever seen something that's got a positive on one end and a negative on the other end? It's like a battery or magnet battery. And so we have a voltage that's going to exist across our membrane because we basically have a biological battery. And that voltage is going to be what we refer to as our membrane potential. Where the positive charges are, where the negative charges are. And it's not going to be static. We're going to have molecules passing back and forth through channels, some of which are leaky, some of which we can control being open or not. And in fact, potassium is going to be one of those leaky ones. It's continually wanting to leak out diffusion following its concentration gradient. And we're going to be using energy, as we're going to see with a pump, that's continually trying to keep sodium out and potassium in. We expend a ton of energy just trying to maintain those concentration gradients. Not to have them just to sit around, but for when we need to accomplish work, we need those steep gradients to make the work happen. And we'll see what that is coming up. So here in our illustration, and this is a new one, again, if you've got the older PowerPoint version. This is extracellular outside the cell, cytoplasmic inside. Inside's going to be more negative because of all those fixed anions. And if you could put an electrode in and measure the difference between the more positives outside, the more negatives inside, you would get a reading for that neuron at rest in the neighborhood of minus 70 millivolts. Write that number down. Minus 70 millivolts is what we will consider the resting membrane potential of a neuron. And in physiology, that's what it's going to be as well. Now there's a range depending on your cell type and depending on where the neuron is and what the neuron's doing. I like simple things. I like numbers with zeros and fives in them. So for us, we're just going to consider the resting membrane potential of all neurons minus 70 until someone that's smarter than us tells us different. Are we good with that? But do you see how we got this number? And do you understand why the number is minus 70? What does that negative 70, what does that mean? We got a potential of 70 millivolts, but the negative. Where do we have more of our negative charges, in or out? Inside. So a negative potential means we have more negative charges inside the cell. If we had a positive 70, that would mean that we have more positive charges inside compared to out. All right? But since we have negative charges on the inside, positive charges on the outside, just like a battery, we call the positive and the negative like posts on your battery for your car. Sometimes we call those the what? Positive and negative. Here on Earth we have a north and a south pole. Because it's polarized. When you have this charge separation, I mean, if it was the same, would we be polarized? What would we be? Unpolarized or neutral, right? Be neutral. So when we have this charge separations, we're said to be polarized. And that's going to be an important concept as we look at changing what our cells are doing. So we see potassium wants to diffuse out following its concentration gradient. Does diffusion require any energy? Nope. Sodium wants to diffuse in. Why? Following its concentration gradient. And who is sodium's best friend? You always see them together as often as they can. Chloride. So chloride's going to diffuse into the cell, and it's following sodium. So we're going to use these concentration gradients to our advantage so that we can send an electrical signal down an axon that may be a meter in length. And it's just going to be the moving of sodium and potassium ions. That's it. But that's how we can send a signal. And we can send that signal of these changes in sodium and potassium at a speed of over 100 meters per second. Is that crazy? That's just crazy. Just this changing these ions across the membrane sends a signal at over 100 meters per second. Here's my summary slide. Outside the cell, inside the cell. The A's with the negatives, those are our fixed anions that can't get out. And you see potassium, we've got way more potassium inside the cell. And out here we've got our sodium predominantly, but we've also got our chloride. And notice down at the bottom, the arrows are pointing to high concentration of sodium. Where? Outside. And where's the potassium high concentration? Opposite. Inside. And chloride simply following the sodium. So there's a lot of information that's going to be very useful for you if you just learn, if nothing else, just learn this bottom stuff down here. Out and in. Anions, fixed anions, because they're negatively charged. You may have some, but predominantly we have the fixed anions, and they dominate, and so that's where we get that negative inside the cell. So here's an illustration, again, showing our resting membrane potential, minus 70. We've got our potassiums inside. We've got our sodiums outside. Here in the middle, we've got this pumping action going on using energy. If there's too many potassium out, we're going to pump them in and at the same time pump sodium out. That's called the sodium-potassium pump. That one pump is used so many places in your body that it accounts for about 12% of your total energy expenditure every day, which blows my mind. But every neuron is going to have a ton of these sodium-potassium pumps maintaining and what we'll see resetting our concentration gradient of sodium high out, potassium high in. But we got to burn energy to make that happen. So we're going to start with this illustration. Now we're going to put it to work. We're going to put it into motion. And our motion is going to be what we see. Here we have an illustration of an axon. The cell body on the left, there's our axon, and we're sending the signal to the right. This is that nerve impulse that goes from your brain to your fingers so you can snap your fingers. And if we could visualize the change of our membrane potential along the length of that axon, do you see this wave of electrical change and how it's sliding down that axon? That change is called the action potential. And so we're going to examine how this wave is able to move. Now, the first thing we're going to do is we're going to pause this image and analyze each part of that graphic. Because we have a part that goes up, a part that goes way down, a part that goes too far, and then we reset and do it again. So we've got three pieces, the up, down, the little dip at the bottom. We've got to understand what's happening in each one of those pieces of the puzzle. Now go ahead and introduce here the change in our membrane potential from our minus 70, our resting membrane potential, that rocket that goes straight up. We're becoming more positive. That's called depolarization. Because if we're going from minus 70, if we're going up towards zero, what did I say zero membrane potential was? Neutral. No polarization. You get the term? Depolarizing. You're removing the polarization as you're getting closer to zero. Even when we go past zero and we're still going up, it's still called depolarization. Depolarization is moving more positive for your membrane potential. Now, if our membrane potential is becoming more positive, what must we be doing? We're depolarizing, but how are we making, if we have a positive membrane potential, what do we have more of on the inside now? Positive ions. So if we want to make the inside of the cell more positive, we can't get rid of the negative charges that are already there. What must we do? We got to let more positives in. What do you think the more positive ion we're going to let in is? Sodium, because we got a lot of it out there. All we got to do is open a gate. The sodium's going to come rushing in. And that gate is what I want to talk about now. Because that gate is what we most often refer to as voltage-gated ion channels. They're these protein channels Much like we saw integral membrane, that you create this sort of barrel or channel or tunnel. And these voltage-gated channels are extremely controllable, I guess if I could make up a word, regulatable maybe. Because when we look at our voltage-gated channels, they can exist in 3 different states. Certainly we got open and closed, right? But there's a third one that's going to be extremely important as we look at our voltage-gated channels. Now, why do you think we call them voltage-gated? Well, when you change the voltage, that's when you open or close them. Like we turn.
Like the membrane where like it can choose?
The membrane can't choose. No.
When the number gets high enough, it automatically just flaps shut.
When the number gets to a certain point, it's going to change. Maybe shut, maybe open. Okay? At rest, at our minus 70 millivolts, our sodium voltage-gated channel is closed. That's one of the states it can exist in. But when we change that membrane potential, and for our sodium voltage-gated channel, it has a sensor built into it, which is a change of amino acid, a change of shape. For our sodium voltage-gated channel, when we reach minus 55 millivolts, so that's that moving upward toward depolarization. When we get to minus 55 millivolts, sodium voltage-gated channel opens. And that's the second state within which we can exist. So we've got open configuration. Materials can pass, but it's going to be selective. For a sodium voltage-gated channel, what's the only thing that's going to pass? Sodium. Same for potassium. We already talked about closed. That's going to be that gate that's going to slam shut. But the gated part, the closing part, takes a little bit longer. So we have a third component. Our voltage-gated channel can open, but before it can close, it can also inactivate. So the only way ions can pass is in an open channel. If that channel is open and inactivated, nothing passes. If it's closed, nothing passes. It's just the inactivation part can happen much more rapidly than it can close again. So here is an illustration of an open channel. There's the little gated part that they have sort of right in the middle. So when you open that, materials can pass. If this is a potassium voltage-gated channel, and if that potassium voltage-gated channel opens in our cell, in the membrane, where is potassium going to go? Think about your answer. The potassium voltage-gated channel in our membrane. If that channel opens, where is potassium going to go? Out, because high concentration inside. Diffusion is going to push it out. So that's our open channel. There, we can close it. You can see we've got the open channel. There it's closed. At rest, our sodium voltage-gated channel, potassium voltage-gated channels, they're both closed. But at minus 55 millivolts, the sodium channel swings open. Not at minus 56, not at minus 58, but minus 55 or more positive, that will open that sodium voltage-gated channel. Now, you see this the illustration here's got this little ball down on the end of it. That's to illustrate another part of the receptor that can cause an inactivation or plugging up the hole in a different way. Here, they don't show the gate, but the gate would be open still. And this ball swings up, blocks the passage, so nothing's going to get through. So for our cell, when we open that sodium voltage-gated channel at minus 55 millivolts, sodium comes rushing in, following its concentration gradient, but almost as soon as it opens, it inactivates. It lets a ton of sodium in really, really quickly because of that humongous gradient. But then, while it's inactivated and nothing else can pass, it's in the process of closing and resetting. Okay? So we've already seen resting membrane potential. We're seeing how we're getting more positive through a stimulus. And again, when you get to physiology, we'll talk all about excitatory and inhibitory stimuli. But we've got to reach that minus 55 to open up the sodium voltage gated channels, allow sodium to come in so that we can go very positive very quickly in the part that we call depolarization. That's the first part of our action potential, depolarization. So you remember our graph that was sliding along the axon? We've stopped it, we've paused it right here and looking at the different parts, and we're going to go through those.
So how does it start going from negative 70 to get to 55?
There are a number of different stimuli that can change that. We're going to see that there are chemical signals that can open other ion channels that allow for a more positive change. If you open up another sodium channel or you open up a receptor that allows a lot of sodium to come in, that's going to start making that more positive. As long as you move it up to minus 55, those sodium voltage gated channels are going to automatically open. Okay? And we got another way we're going to do that as far as along the length of the axon. Once you start it at this end, you can't stop it. And we'll talk about that as we get to the back part. So there's our threshold, that's threshold for excitation, minus 55 millivolts when you open the sodium voltage gated channel. Once you do, you can't stop it. It's going to go to completion. It's the all or none rule. You get to minus 55 or you don't. You have an action potential or you don't. That's it. There's no gray area with an action potential. So our depolarization happens because we open our sodium voltage gated channels. But as soon as we open them, our sodium voltage gated channels are going to become inactivated. But that's still enough for us to go all the way up, upwards of positive 30, positive 40 millivolts. But on the way up, we're also going to open potassium voltage gated channels. Now potassium is going to start going out, which makes the inside more negative, right? That's going to lead to this downward slope, which we call repolarization. You're trying to reestablish resting membrane potential or reestablish the polarized state of the membrane, and that's the downward slope based on potassium going out. And the potassium voltage gated channel flashes open and inactivates, but it can't inactivate so quickly that we don't overshoot down at the bottom. Do you see how we get more negative than resting membrane potential? So we're even more polarized, and that last bit where we're too negative, that's called hyperpolarization. Extreme polarization, too much polarization.
So the low lines, that little loop right there is hyperpolarization.
This little loop right there, that's hyperpolarization. And the reason that it comes back up to resting membrane potential is our sodium-potassium pump reestablishing, moving the potassiums back in, moving the sodiums back out, reestablishing the gradient, and at the same time, reestablishing our resting membrane potential so we can do this again, and again, and again, and again. Okay, we're going to go through each one of these steps. This is just kind of an overview, okay? If we don't get it by the end, just let me know. So, action potential starts in a place and it moves down our axon, alternating sodium in, potassium out. That's what makes the wave. That's what makes it move down the axon. There's our little sodium-potassium pump working overtime to reestablish those gradients. So, resting membrane potential, we have our sodium and potassium channels closed. Minus 70 millivolts. The first thing is that depolarization, right? Opening the sodium voltage gated channels, sodium comes flooding in. At what voltage? Minus 55 millivolts. That's the threshold for sodium voltage gated channel. That's also the threshold to start an action potential. If you don't have a sodium voltage gated channel in the membrane of that cell, it will not undergo an action potential. period. You can change the membrane potential all day long. It will never be an action potential. So here in our other illustration, the upward arm getting more positive, depolarization, the opening of the sodium voltage gated channels. But remember what happens almost as soon as you open them? The inactivation, before we get to the potassium. As soon as you open the sodium channel, it inactivates. But it can't do it fast enough not to let enough sodium in to go through the roof. Repolarization, that's going to be the opening of potassium channels. We've got the sodium channels inactive, no sodium's moving, but now the potassium starts coming in. Now understand this as well. When our sodium channels are inactivated, are we ever going to be able to let any more sodium back in while they're inactivated? No, the answer is no. So even if we were to have another stimulus right behind the first stimulus, there was a very powerful stimulus that could change our membrane potential to minus 55 or more positive, you cannot start another action potential while our sodium voltage gated channels are inactivated. So that period of inactivation of our sodium voltage gated channels is referred to as the absolute refractory period. You can't have another action potential. I don't care. It's absolutely not going to happen. And it's because of the inactivated state of our sodium voltage gated channel. Now we said we open potassium voltage gated channels. Potassium goes where? Potassium goes out, makes the inside more negative, and that's going to be our repolarization. See, there's our potassium out. Notice, sodium voltage gated channel, it's open, but it's inactivated. Sodium's not going anywhere. Now potassium goes out, and that's going to start to drop our membrane potential, and it's this downward arm that's coming down because of our potassium leaving the cell. And again, along the top, you can see our absolute refractory period happening sort of in the middle. Absolute refractory period because the sodium voltage gated channels are inactive. So that's the repolarizing phase because of potassium leaving the cell. Now again, we inactivate the potassium voltage gated channel, but not before we've allowed enough potassium to leave that we're going to overshoot. And that overshoot part is the hyperpolarizing phase. So again, still letting because we let potassium out, we get more negative, and our little dial should be it should be pegged down here at the floor on the left side because we're going to be minus 90 instead of minus 70. That's the hyperpolarizing part. Now, during the hyperpolarized period, that's when we possibly could have another action potential, but the stimulus to get it started is going to have to be a lot stronger. Because now, instead of going from minus 70 to minus 55, what changes that? Is that 15 millivolt change? We got to go from 90 to minus 55, so a 35 millivolt change. You see how it's got to be stronger? And if you had a stronger stimulus, you could start another action potential because in our hyperpolarized period, our sodium voltage gated channels have reset and they're closed and they could be opened again. So we could have another. It's just going to have to be a really, really strong stimulus to get us from minus 90 or wherever we're going to be upward to minus 55.